10 fascinating facts about human DNA. The structure of DNA, the secret of the double helix. The human body is one of nature's most complex creations. Inside every cell lies information that governs life, determines the future, and passes from generation to generation. The main code of this mysterious information is DNA, or deoxyribonucleic acid. Although science has explored this molecule in depth, many of its secrets remain undiscovered. In this section, we will examine the most important feature of DNA, its double helix structure. 1. The discovery of DNA. The first information about DNA appeared in the 19th century. In 1869, Swiss scientist Friedrich Miescher studied white blood cells and isolated a special substance which he called nuclein. Later it was discovered that this substance resided in the nucleus of the cell. However, its exact function remained unknown for a long time. By the mid-20th century, scientists realized that DNA was the main molecule responsible for heredity. In 1953, British scientists James Watson and Francis Crick, using X-ray crystallography data, revealed the double helix structure of DNA. This discovery sparked a revolution in biology. 2. The meaning of the double helix. Simply put, the double helix is made up of two strands of DNA twisted around each other, resembling a coiled ladder. Each rung of the ladder consists of special chemical substances known as nitrogenous bases. There are 4 types of nitrogenous bases in DNA: adenine, A, thymine, T, guanine, G, cytosine, C. The pairing is very specific. Adenine always pairs with thymine, AT, guanine always pairs with cytosine, GC. This rule is known as the complementary base pairing principle, which ensures stability and accurate replication of genetic information. 3. Stability of the structure. The double helix is not only elegant in shape, but also essential for the durability of the molecule. Hydrogen bonds between the bases, the rigidity of the phosphate backbone, and the helical coiling all contribute to the DNA's stability. This is why our genetic information can be faithfully transmitted from cell to cell and from parent to child. 4. Functions of the double helix. The double helix structure is not just a chemical form, it serves as the foundation of life, performing several key functions. Information storage. DNA carries instructions for making every protein in the body. Each protein is coded by a specific gene. Replication. Before a cell divides, the DNA strands separate and each acts as a template for a new complementary strand. This results in 2 identical DNA molecules. Mutation. Sometimes errors occur in the replication process. These mutations can be harmful, beneficial, or neutral, and they play an important role in evolution. 5. Scale of DNA. The scale of DNA is astonishing. If you stretched out the DNA from the nucleus of a single human cell, it would be about 2 meters long. If you combined all the DNA in the body, its length would exceed the distance from the Earth to the Sun. How such immense information fits into a tiny nucleus remains one of nature's wonders. 6. Scientific and medical significance. The discovery of the double helix opened a new era in genetics and has many applications today. Medicine, diagnosing genetic predispositions and diseases. Forensics, identifying criminals through DNA profiling. Archaeology, studying ancient remains to trace ancestry. Biotechnology, developing new medicines and vaccines through genetic engineering. 7. Symbolism of the double helix. Today, the double helix of DNA is more than a scientific concept. It has become a symbol of life, progress, and the future. It appears in logos, artworks, and even jewelry designs, representing the essence of living systems. The double helix is one of the greatest scientific discoveries in human history. It elevated not only biology, but also science as a whole. By understanding the structure of DNA, we uncover who we are, how we have evolved, and what possibilities lie ahead. In one simple molecule, the destiny and history of humankind are written. The length and weight of DNA. The human body consists of trillions of cells, and each cell contains DNA, which stores genetic information. One of the most astonishing facts about DNA is its length and weight. At first glance, DNA is invisible to the naked eye, yet it spans unimaginable scales. In this section we will explore the measurements, volume, and fascinating features of DNA. 1. Length of DNA in a single cell. Each human cell nucleus contains about 2 meters of DNA. How can such a long molecule fit inside a tiny nucleus only a few micrometers wide? The answer lies in the DNA's remarkable packaging ability. DNA is tightly wound around special proteins called histones, forming structures known as nucleosomes. These nucleosomes coil further, ultimately forming chromosomes. This complex packing allows meters of DNA to fit into a microscopic nucleus. 2. Total length of DNA in the human body. On average, the human body has about 37 trillion cells. If the DNA in each cell were stretched out and placed end to end, the total length would circle the Earth millions of times or travel back and forth between the Earth and the Moon thousands of times. According to scientists, the combined length of all the DNA in the human body exceeds 100 billion kilometers, reaching to the very edge of the solar system. 3. The weight of DNA. The weight of DNA is equally fascinating. The DNA in a single cell weighs only a picogram, 1/1,000,000th of a gram. But when all the DNA in the human body is combined, its weight is roughly 70 grams, about the weight of a bar of chocolate. In other words, the physical mass of the code that defines our entire existence is no heavier than a small sweet. 4. The storage capacity of DNA. Beyond its length and weight, DNA's storage capacity is astonishing. Researchers estimate that 1 gram of DNA can store about 215 petabytes of information. This is enough to hold millions of movies or even all the data on the modern internet. For comparison, today's hard drives typically store only a few terabytes, making DNA billions of times more efficient. 5. Viewing DNA under the microscope. DNA cannot be seen with the naked eye, but under a microscope it can be observed in the form of chromosomes, the most compact packaging of DNA. Humans have 46 chromosomes, each containing millions of nucleotides. The precise sequence of these nucleotides determines everything from our body shape and eye color to aspects of our personality. 6. Similarities and differences. Human DNA is 99.9% identical across individuals. The remaining 0.1% accounts for all the differences we see in humanity. Height, skin tone, eye color, temperament, and countless other traits. That tiny fraction gives rise to the immense diversity of human life. The number of genes in DNA and their role. Human DNA is not just a long thread, it is a complete instruction manual. Within its structure are segments that determine how our bodies function and what characteristics we inherit. These segments are known as genes, the fundamental units of life. 1. What is a gene? A gene is a section of DNA that codes for the production of a specific protein or RNA molecule. Proteins are the workhorses of the cell. They form enzymes, build hormones, defend the body, and make up structural components of tissues. In other words, genes are the codes that run our biological program. 2. How many genes do humans have? After sequencing the human genome, scientists discovered that humans have about 20,000, 25,000 genes. Earlier estimates suggested hundreds of thousands, but it turned out that most of our DNA does not consist of genes. Interestingly, the number of human genes is not significantly higher than in some simpler organisms. Rice, for example, has about 40,000 genes. This does not mean humans are less complex. Complexity depends not on the number of genes but on how they are regulated and interact. 3. Functions of genes. Genes play diverse and vital roles. Protein synthesis. Each gene contains instructions for building a protein. Growth and development. Genes regulate the formation of bones, muscles, and nerves. Physiological regulation genes control processes such as hormone production. Defense genes shape the immune system. Behavioral influence. Some genes affect psychological and neurological traits. 4. Genome and junk DNA. Only about 1.5% of human DNA actually consists of genes. The rest was once called junk DNA. However, recent studies show that these non-coding regions play crucial roles in regulating when and how genes are activated. This is the focus of a growing field known as epigenetics. 5. Genetic disorders. Mutations or errors in genes can lead to diseases such as cystic fibrosis, hemophilia, muscular dystrophy. This makes genetic research critical for medicine. Today, scientists are developing gene therapy, which aims to repair or replace defective genes to treat genetic disorders. 6. Genetic diversity. Genes make each human unique. Although human DNA is 99.9% identical across individuals, the remaining 0.1% accounts for differences such as eye color, hair type, height, and susceptibility to certain diseases. Genes are the smallest yet most essential elements of life. They define everything about us from appearance to physiology. By studying genes, humanity gains deeper insight into its origins, health, and future. DNA similarities among humans. Although humanity appears diverse, our DNA reveals a profound truth. We are far more alike than different. Despite variations in height, skin tone, culture, and lifestyle, on a genetic level we all belong to one extended family. 1. Level of similarity. According to genetic research, the DNA of all humans is 99.9% identical. This means that a person from Japan, Africa, America, or Kazakhstan shares almost the exact same genetic code. The remaining 0.1% difference accounts for individuality such as skin color, hair texture, eye color, susceptibility to certain diseases, body height and build, even aspects of personality. It is this small fraction that creates the incredible diversity of humankind. 2. Why are we so similar? The primary reason lies in our shared ancestry. Genetic studies trace modern humans back to a relatively small population that lived in Africa about 200,000 years ago. Thus, no matter how different we may look on the surface, genetically we are relatives. 3. Comparison with other living beings. DNA similarity extends beyond humans. Humans and chimpanzees share 98–99% of their DNA. Humans and mice share about 85%. Surprisingly, humans and bananas share nearly 50% of their DNA. These comparisons highlight the interconnectedness of all life on Earth and support the theory of common evolutionary origins. 4. Scientific importance. Understanding human DNA similarity is vital for science. It plays a role in medical research, identifying genetic disorders, developing effective treatments. For example, a drug tested in one part of the world often works globally because the genetic foundation of humanity is nearly the same everywhere. The fact that human DNA is 99.9% identical is a powerful reminder that we are all part of one human family. Our differences may seem large, but they arise from only 0.1% of our genetic code, a small fraction that makes each of us unique. Human DNA compared to animals, similarities and differences. Humans share a common evolutionary origin with animals, which is why much of our DNA is similar. However, there are also crucial differences that make humans unique. 1. Similarity with chimpanzees. Our closest relatives are chimpanzees. Human DNA is 98–99% identical to chimpanzee DNA. Similarities: bone structure, muscle system, parts of the immune system. Differences: brain size and cognitive abilities. The human brain is 3 times larger and capable of advanced reasoning, creativity and language. 2. Comparisons with other animals. Macaque monkeys about 93% similarity. Cats nearly 90% similarity with humans. Mice about 85% similarity. This is why they are widely used in medical research. Dogs about 84% similarity. Bananas, surprisingly humans share about 50% of DNA with bananas. 3. Key differences. The major differences between humans and animals include brain and cognition. Humans have a highly developed cerebral cortex. Language and speech: humans possess unique genes, like FOXP2, that enable complex speech. Social complexity: humans build cultures, arts and sciences. Longevity: humans generally live longer and have advanced healthcare. 4. Scientific significance. DNA similarities with animals are crucial for studying diseases, testing medicines, understanding evolutionary processes. While human DNA is highly similar to that of animals, the subtle differences are what set us apart. These differences underpin our ability to think critically, speak, and develop culture and civilization. Mutations in DNA and their effects. DNA is a highly complex and delicate structure containing billions of genetic letters. However, this code is not always stable. Any change in the DNA sequence is called a mutation. 1. What is a mutation? A mutation is an alteration in the DNA sequence. It can range from a single nucleotide change to the loss, duplication, or insertion of large DNA segments. 2. Types of mutations. Point mutation. A single letter is substituted, for example A instead of G. Deletion. A portion of DNA is lost. Insertion: extra nucleotides are added. Duplication: a DNA segment is repeated. Chromosomal mutation: changes in the number or structure of entire chromosomes, for example Down syndrome caused by an extra copy of chromosome 21. 3. Causes of mutations. Natural errors. Mistakes during DNA replication and cell division. External factors: ultraviolet radiation, ionizing radiation, chemical exposure, viral infections. 4. Effects of mutations. Mutations can have various consequences. Harmful mutations cause genetic disorders, for example, hemophilia, thalassemia. Neutral mutations have no significant impact. Beneficial mutations sometimes provide an advantage. For example, certain mutations make individuals resistant to HIV infection. 5. Role in evolution. Mutations are the driving force of evolution. Without them, life would not adapt or change. Every new trait or adaptation originates from mutations, later preserved by natural selection. 6. Importance in medicine. By studying mutations, medicine can detect genetic diseases early, provide genetic counseling, develop gene therapies. For example, CRISPR-Cas9 technology allows scientists to correct certain harmful mutations. Mutations are a double-edged sword, sometimes harmful, sometimes beneficial. They influence human health while also serving as the foundation for evolution and diversity in life. The link between DNA and personality traits. A person's character, abilities, and personality are influenced not only by upbringing and environment but also by the genetic code embedded in DNA. This field of study is known as behavioral genetics. 1. Genes and behavior. Some genes are known to influence human behavior. DRD4 gene, linked to the dopamine system. Certain variants are associated with novelty-seeking and adventurous personalities. MAOA gene, sometimes referred to as the warrior gene, influencing aggression levels. SLC6, a 4-gene serotonin transporter gene, linked to susceptibility to stress and depression. 2. Heredity and environment. Twin studies help reveal the balance between genetics and environment in shaping personality. Identical twins, sharing 100% of genes, often display strikingly similar traits, even when raised apart. This demonstrates the role of genetics. However, environment also plays a critical role. Upbringing, culture, and social context shape personality development. 3. Genes and abilities. Genes also influence certain abilities: musical aptitude, quick problem-solving, athletic potential. Yet these natural inclinations require effort, practice, and training to fully develop. 4. Genetic predisposition. Genes may predispose individuals to certain traits or conditions, but they do not guarantee outcomes. For example, a person may carry obesity-related genes but stay fit with healthy habits. Someone may carry genes linked to depression but remain mentally resilient in a supportive environment. DNA influences personality, but it does not dictate destiny. Genes provide possibilities, while environment, personal choices, and determination shape who we ultimately become. The history of DNA research and discoveries. The study of DNA represents one of the greatest revolutions in human knowledge. These discoveries transformed not only biology and medicine, but also our entire understanding of life. 1. Early steps. 1869. Swiss scientist Friedrich Miescher extracted a unique substance from white blood cells and called it nuclein. It was later identified as DNA. Early 1900s. Scientists introduced the concept of genes to explain inheritance, but the connection to DNA was still unknown. 2. Development of genetics. Gregor Mendel's work, 19th century, revealed the mathematical laws of inheritance. 1920s. The link between chromosomes and genes was established. 3. Discovery of DNA structure. 1953. James Watson and Francis Crick unveiled the double helix structure of DNA, one of the greatest scientific breakthroughs of the 20th century. Their work was supported by Rosalind Franklin, whose X-ray crystallography images provided critical evidence. 4. Decoding the genetic code. In the 1960s, scientists discovered that each set of 3 nucleotides, codon in DNA, corresponds to a specific amino acid. This cracked the code of protein synthesis. 5. The Human Genome Project. 1990-2003, the Human Genome Project was launched. It successfully mapped all human genes, about 25,000. This milestone opened new frontiers in medicine: personalized diagnostics, gene therapy, and biotechnology. 6. Modern advances. CRISPR-Cas9 technology allows precise editing of DNA. Artificial intelligence and big data accelerate genetic discoveries. Commercial genetic tests now trace ancestry and identify predispositions to diseases. The history of DNA research reflects humanity's relentless pursuit of knowledge. From the first discovery of nuclein to genome editing, DNA science has transformed how we understand ourselves and holds the key to the future of medicine and evolution. DNA in ancestry research and forensic science. DNA is not only central to medicine but also a powerful tool in history, anthropology, and law enforcement. It helps us trace our origins, map human migration, and solve crimes. 1. Ancestry research. Genetic testing allows people to uncover their ethnic composition and ancestral roots. Y-chromosome DNA traces paternal lineage, while mitochondrial DNA, mtDNA, traces maternal lineage. These tests can show where a person's ancestors lived thousands of years ago. 2. Human migration studies. Specific DNA markers help track the migration of ancient populations. Genetic research confirms that all modern humans originated from Africa tens of thousands of years ago. 3. Public interest. Today, direct-to-consumer DNA tests are widely available. Within weeks, individuals can receive reports about their ancestry and even health-related genetic traits. 4. DNA in forensics. In criminal investigations, DNA serves as a genetic fingerprint. Since each person's DNA is unique, traces left behind, hair, blood, saliva, or skin cells, can identify suspects. The first forensic use of DNA profiling occurred in the UK in 1986. 5. Identifying the missing and historical figures. DNA helps identify unknown remains, missing persons, and victims of war or disasters. It is also used to confirm the identities of historical figures and uncover details about their ancestry and health. DNA is a bridge between past, present, and future. It tells us who we are, secures justice in society, and continues to shape the way we understand humanity. The future role of DNA in science and conclusion. DNA is one of the most powerful keys in modern science. It not only unlocks our past but also shapes the future of medicine, technology, and society. Many of the greatest scientific revolutions of the 21st century will revolve around DNA. 1. Gene therapy. In the future, the primary treatment for genetic diseases will be gene therapy. By replacing faulty genes with healthy ones, scientists hope to prevent or cure inherited disorders. Early trials have already shown promise in conditions like hemophilia and immune deficiencies. 2. CRISPR and gene editing. The CRISPR-Cas9 system works like molecular scissors that cut and edit DNA. It holds the potential to eliminate diseases, create new medicines, and even grow artificial organs. However, ethical questions remain. How far should we go in altering human genes? 3. Personalized medicine. By analyzing an individual's DNA, doctors will be able to design treatments tailored specifically to each patient. A drug that works for one person may not work for another. In the future, medicine for each patient will replace the one-size-fits-all model. 4. DNA as a data storage system. DNA is not only a biological molecule but also a data carrier. Scientists have already stored text, images, and even videos in DNA molecules. Its capacity is astonishing. 1 gram of DNA can store all the world's internet data. 5. Agriculture and ecology. DNA technologies can improve crop yields, create pest-resistant plants, and support food security. Genetic tools also help preserve endangered species by maintaining their genetic diversity. 6. Ethical and social challenges. While DNA science offers limitless possibilities, it also raises concerns. Issues such as genetic discrimination, privacy of personal data, and the possibility of designer babies spark debate worldwide. Conclusion. DNA is the ultimate blueprint of life. It reveals our origins, helps us fight diseases, and guides humanity into the future. The study of DNA is not only a triumph of science but also a responsibility, shaping the destiny of civilization itself. DNA is the ultimate blueprint of life, revealing our origins, helping us fight diseases, and guiding humanity into the future, shaping the destiny of civilization itself.